In this video, we will talk about glycolysis. Glycolysis is a metabolic pathway in which a glucose molecule is broken down and converted into two molecules of pyruvate or pyruvic acid and produces energy in the form of ATP, that is adenosine triphosphate. So, where does glycolysis occur? As this is the first step of energy production in our body, it occurs in all the cells of the body. Glycolysis takes place outside the mitochondria and inside the cytoplasm of the cells. Depending on the availability of mitochondria and oxygen in the cell, the process of glycolysis can be divided into two types: the aerobic glycolysis and the anaerobic glycolysis. In the both types, the starting material is the glucose and the end product is pyruvate. However, the results differ in the number of ATP production. When mitochondria and oxygen is available in the cell, the pyruvate that produced in the cytoplasm enters into mitochondria and starts Krebs cycle, that is another metabolic pathway, and a lot of energy in the form of ATP is released. But when mitochondria or oxygen or both are not present in the cell, the pyruvate enters into lactic acid cycle, which occurs also in the cytoplasm and produce ATP, but that is much less compared to aerobic process. I'll let you know in detail about the Krebs cycle as well as lactic acid cycle in later videos. So aerobic can take place in most of the cells of the body, but anaerobic takes place in cells where there is lack of mitochondria, such as erythrocytes, cornea, or lymphs. Now let us see the steps or reactions involved in the process of glycolysis. One glucose is converted into two pyruvates, but there are 10 reactions in between. Whenever a glucose molecule enters into the cell, the first reaction of glycolysis starts. This is the structure of a glucose that has a 6-carbon ring chain. Let's imagine the carbon chain as this. In the first step, glucose is converted into glucose 6-phosphate. This is a phosphorylation reaction. In this step, one ATP is converted into ADP. It means one energy has been utilized here. A phosphate from this ATP is attached to the 6th number of carbon of glucose molecule and the ADP is released. The step takes place by the enzyme kinase. There are two types of kinases enzymes at this stage, which are operated by the concentration of glucose. Hexokinase: if glucose concentration inside the cell is in little amount, the hexokinase enzyme is activated. But if the concentration of glucose inside the cell is high, the glucokinase enzyme becomes active. Now, another important thing in this step is this is an irreversible reaction. Once the glucose is converted into glucose 6-phosphate by the kinase enzyme, it cannot convert back to glucose by the same reverse process. That's why the reaction is irreversible. Now step 2. This is an isomerization reaction. Glucose 6-phosphate is isomerized and produce fructose 6-phosphate. Fructose is also a 6-carbon chain structure. The enzyme functions here is a phosphoglucose isomerase, and the reaction is a reversible reaction. It means the same enzyme can reverse the reaction from fructose 6-phosphate to glucose 6-phosphate. It is showed like this. Now step 3 is another phosphorylation reaction where another ATP is utilized. Fructose 6-phosphate is converted into fructose 1,6-bisphosphate. It seems one ATP is utilized again and the phosphate is attached to the number 1 carbon of fructose 6-phosphate, and thus now the molecule has 2 phosphate group at the two ends, and the enzyme here is phosphofructokinase 1. This is also an irreversible reaction. One important thing here is some of the fructose 6-phosphate will be taken by the another enzyme, phosphofructokinase 2, and that is fructose 6 so fructose 6-phosphate will be converted into fructose 2,6-bisphosphate instead of fructose 1,6-bisphosphate. Why is that? When there are high concentration of glucose present in the cell, it means the process should run very faster. So concentration of fructose 6-phosphate will also be high. In this step, phosphofructokinase 2 enzyme will also be activated and produce fructose 2,6-bisphosphate, and the production of fructose 2,6-bisphosphate will raise a signal and stimulate the phosphofructokinase 1 enzyme. The extra stimulation of this enzyme will make the glycolysis process even faster by making the reactions moving forward. So this is a very important step of glycolysis, as well as the enzymes are also important. We'll discuss about these enzymes in a video later. Now, in the fourth step, fructose 1,6-bisphosphate is splitted in the middle and 2 molecule is produced here. You see, 1 molecule with 6 carbon chain is splitted in the center and 2 molecules are produced with 3 carbons in each one. This is dihydroacetone phosphate, DHAP, and this is glyceraldehyde 3-phosphate, G3P. Now, DHAP contains a phosphorylation at carbon 1 point and G3P contains phosphorylation at carbon 3 point. The enzyme works here is aldolase and the reaction is reversible. The fifth step is nothing but the interconversion between DHAP and G3P. This is also a reversible reaction, but as only G3P moves forward in the glycolysis process, the DHAP keeps converting itself into G3P. As a result, 2 molecules of G3P is finally produced. The enzyme is triose phosphate isomerase here, and this is also a reversible reaction. Now, in the sixth step, both this G3P is converted into 2 molecules of 1,3-bisphosphoglycerate by the enzyme glyceraldehyde 3-phosphate dehydrogenase. This enzyme is very complex enzyme and carry 3 domains within it. 1 inorganic phosphate, 1 NAD+, that is nicotinamide adenine dinucleotide. This is a coenzyme, and 1 empty space for capturing the G3P. So what actually happens here? With this enzyme, G3P is actually phosphorylated with the inorganic phosphate, but no ATP is utilized in this step like in step 1 and 3. 1 NAD plus is converted into NADH plus and lots of energy is trapped in this way. Okay, don't get confused. NADH plus will be producing more ATP through electron transport chain. I will make another video on this topic. Right now, just believe that a lot of energy is trapped here, okay? So here actually 2 NADH plus is produced as both the G3P are converted. And one more thing. This is the first oxidation reaction of the glycolysis process, and this is a reversible reaction. Step 7 brings the first dephosphorylation reaction of the process, which is a reversible reaction where phosphate group will be released from the molecule. You see, 1,3-bisphosphoglycerate is dephosphorylated and converted into 3-phosphoglycerate. The phosphate group from number 1 carbon is removed by the enzyme phosphoglycerate kinase. Now, the removed phosphate group is attached with 1 ADP and produce ATP. So as 2 molecules of 1,3-bisphosphoglycerate are converted, thus 2 ATPs are produced. This is a reversible reaction. Step 8 is an isomerization reaction where 2 molecules of 3-phosphoglycerate is converted into 2 molecules of 2-phosphoglycerate. Now the phosphate group has just changed its place from number 3 carbon to number 2 carbon. The enzyme is phosphoglycerate mutase here, and this is also a reversible reaction. Now, 2-phosphoglycerate, this is converted into phosphoenol pyruvate in the 9th step. The enzyme here is enolase. This is a reversible reaction. This is just the preceding step of final one. Here also 2 molecules are produced. And in the 10th and final step, 2 molecules of this phosphoenol pyruvate is converted into 2 molecules of pyruvate by the enzyme pyruvate kinase. Just like step 7, the phosphate group is dephosphorylated here and 2 molecules of ATP are produced. The last reaction, where pyruvate is produced, is an irreversible reaction. It cannot convert back to its previous place. So finally, 1 molecule of glucose has converted into 2 molecules of pyruvates by glycolysis process. Now, let us see the gist of this reaction. I mean, all the 10 steps. If we see, we'll find something interesting. There are 10 reactions in which 3 reactions are irreversible, number 1, number 3, and number 10, and the rest of the 7 reactions are reversible. In these 10 steps, 10 different enzymes are activated and do their specific functions. The whole process can be divided into 3 phases. From step 1 to step 3 as phase 1, where 2 molecules of previously available ATP were utilized from the body. Now, step 4 to step 5 as phase 2, where the 6 carbon chain molecule was cleaved into 3 carbon chain molecules. And from step 6 to step 10 as phase 3, where NADH plus and ATP started to be generated. Now, if it is summarized, in phase 1, 2 molecules of ATPs were invested. And in phase 2, the investment were splitted. And in phase 3, total of 4 molecules of ATPs were generated, as well as 2 molecules of NADH were generated, where lots of ATPs are trapped. Now, if we take the example of a business, first we need to invest, then we need to expand the business, and finally make profit. So phase 1 can be named as the energy investment phase, phase 2 as cleavage phase, as the business here is making more branch, and phase 3 can be called as energy generating phase. So this is the beautiful process of glycolysis, through which 1 glucose molecule is converted into 2 molecules of pyruvate and produce ATP in this process. Now the question remains, what happens to this pyruvate and the ATPs and the NADH+. In the part 2 of this video, I'll show you the next interesting steps of energy production. Stay connected and subscribe to this channel for the latest videos. Thank you.